In previous video, we discussed reciprocal cross and monohybrid cross laws. Monohybrid cross laws. So now we are discussing Mendel's analytical and empirical approach. Why Mendel chose two pairs of contrasting characters? So it is a logically one. So the factors are exist in this appearance only. At the time of F2 generation, it produced an odd or heterozygous. That means dominant characters are appear. But F1 generation, dominant characters are appears in F2 generation, and also for this recessive characters or it will be appears, but F1 generation, that recessive traits, it will be hidden, that means disappears, that means disappear. So that is the analytical approach we will call it. Okay, so Mendel's Mendel asked this for 1 by 4 of this that gametes or these characters, it will be hidden. It will be hidden or masked only to reappear in 4 by 1 by 4 of the F2 generation. So it will be concluded that so heterozygous is condition is segregated from the gametes are randomly segregated. The gametes are randomly segregated. So it is that quantitative analysis, in the quantitative analysis the scientists are used that analyzed for this quantitative levels, that analyzed quantitative level. So Mendel's analytical approach is that meticulous one. So in breeding experiments, disintegrate the particular units of heredity present day. Okay. So the analytical approach is truly outstanding one, truly outstanding one. Now they are called as, we are called as genes. First we called as the Mendel's is factors. The rationally is called as the empirical approach. Mendel's experiments were well planned to determine the relationship with the heredity. Well planned stretches are presently executed breeding experiments propose the disintegrate particulate units the heredity. They are transmitted from one generation to the other. Now they are called as genes. So well planned of this determine this heredity traits. So this is traditionally we called out empirical approach. So analysis, what analysis is a quantitative. Okay, quantitative analysis is followed by this in the Mendel's. Next Mendel's analytical approach is empirical. Empirically means Mendel's experiments were planned rationally which govern hereditary traits. That is called this empirical approach. So next we discussed about this test grass. Test grass means the grass is test grass is grassing on individual of unknown genotype with homozygous recessive. Unknown genotype, don't know for this genotype. Test grass is grassing an individual of unknown genotype with homozygous recessive. So Mendel's monohybrid cross, all are F1 generation, all are tall. So F2 generation dwarf and tall plants are here. The ratio is 3:1. So this will be the F1 generation is self-pollinated. F2 generation called dwarf plants as well as tall plants. So and continuously for this F2, F3, F4 generations. So he concluded this genotype of dwarf plants was homozygous. The genotype of tall plants capital T or small t capital T from F1 and F2 cannot be predicted, but all this are homozygous or heterozygous. To determine genotype of tall plant is the Mendel segregated from this apparent plant. So எதுலேருந்து வந்துச்சுன்னு we don't know. நமக்கு எதுலேருந்து வந்துச்சுன்னே தெரியாது. So at the time we will apply on this test cross. This test cross is called all the progenies can be this. All the progenies எப்படி வரும் test crossல? All the progenies recessive individuals. Yeah, it will be crossed with recessive individuals. Okay, so this is the typical test cross organism showing the dominant phenotypes is crossed with recessive parent. Okay, self-crossing, recessive, self-crossing dominant parent. So, you understand the test cross are here. This is the heterozygous test cross. So, parent phenotypes. First one, capital T, small t. Homozygous dwarf plants. So, when the parents are crossed with recessive genotype, that is called as test cross. By the crossing, an organism having unknown genotype. The genotype is not known. Okay, unknown genotype with a homozygous recessive organism. With a homozygous recessive organism. Homozygous recessive organism. So it is possible to know unknown genotype with one generation. Unknown genotype with one generation. So for example, to know whether the given the tall plant has a genotype capital T or capital T small t. Okay, so this genotype first we called out of this heterozygous tall. Heterozygous tall means capital T small t. So whether the genotype is capital T that is the dominant one. So but it is tall. So it crossed with homozygous dwarf. Homozygous dwarf means that is small t. So if the unknown genotype is capital T, this one don't know. Okay, capital T, small t. So the offsprings could be tall. The offsprings could be tall. This one it is. Offsprings could be tall. So definitely you know that. So one, parent one, it is tall. Parent one கண்டிப்பா என்ன செஞ்சிருக்கணும் டாலா இருந்தால் தான் இங்கேயும் டாலாக வரும் அப்படிதானே நம்ம we are crossed with this recessive one. ஓகே recessive one கூட நம்ம வந்து என்ன செய்யும் கிராஸ் பண்ணுறோம் அப்போ நமக்கு தெரிஞ்சிடும் இந்த ஜீனோடைப் என்னவா இருந்திருக்கலாம். Okay, the unknown genotype is capital T, small t and small t. So 50% it be for that in the parent plant. So 50% என்ன இருந்துக்கணும் டாலா இருந்துக்கணும் 50% dwarf இருந்துக்கணும். So we will conclude that 50% of offsprings could be tall, 50% dwarf, 1:1. So the test cross is crossed with this, that ratio is 1:1. So if the cross is made between F1 heterozygous of two factors and the recessive parent, recessive for both factors, it will be given the phenotypic ratio is 1:1:1:1:1. So are you understand? This one is crossed with genotypic ratio is 1:1 and phenotypic ratio is 1:1. So this is the test cross homozygous tall. Yeah, homozygous tall. This is the capital T and but crossed with this recessive parent. Crossed with this recessive parent. Recessive parent. So you understand? This is the crossed with the offsprings are this capital T, small t, capital T, small t. So the same for this 1:1 ratio. 1:1 ratio. This is the ratio. So in that test grass, so we will obtain for this genotypic ratio and phenotypic ratio. We will give this 1:1. So next we discussed about this back cross. So molecular explanation, there is no need. So back cross. Back cross means F1 generations of the hybrid, any one parental genotypes. Backcross will be two types, dominant backcross and recessive backcross. Because the gametes are this two types, dominant and recessive, that backcross is it crossed with this parental. Okay, a cross F1 generation, it will be crossed with parental one, but test cross is crossed with recessive parent only. Only recessive parent, but backcross is crossed with this parental character. The F1 hybrid crossed with parental character. So two types are here, dominant backcross and recessive backcross. So it involves cross F1 offerings either of two parents, either of this two parents. The F1 offsprings are crossed with dominant parents. So the F1 generation, you know that is heterozygous stall, is crossed with this. So dominant parents means capital T, capital T. And to receive the individuals obtain this progeny, what will happen? Both are tall plants. So F1 hybrid is crossed with recessive parents individuals. Both phenotypes appear as equal proportion. So it is specified as test cross. In the recessive backcross helps to identify the heterozygoty of this hybrid. Heterozygoty of this hybrid. So do you know this the backcross when non-individual is crossed with parent? When non-individual is crossed with parent. When non-individual is crossed with. When on individual is crossed with. Individual is crossed with the parent. With the parent is called this backcross. Is called backcross. So a cross between F1 individuals and one of the parents that produce the term is called as backcross. The backcross is used by the breeders to bring useful characters in homozygous condition. It was devised by Mendal. Okay. So for example, backcross. For example, parents. A capital T into small t. What happened? The F1 generation is capital T, small t. So backcross means with the dominant characters it will be crossed. What will? Capital T. So homozygous tall and heterozygous tall, all tall. F1 generation of tall. Okay. So that is the backcross. That is the backcross. F1 generations it crossed with parent. Maybe dominant, that is dominant backcross, and maybe recessive, then recessive backcross.